Let's move on to the next chapter today. It's the anatomy of the flowering plants. Anatomy என்ன உள்ளுக்குற இருக்கிற எல்லாமே தெரியுது தான். For example, hand morphology is just an external appearance. We have all these palm, fingers, all these external things. Anatomy is you will remove the first layer and see what it is. Remove the second layer, what it is. What are the arteries? What are the nerves? What are the bones? What are the things all? So we are going to see that. For that, we should start with tissue and tissue system. We should start with tissue and tissue system. Tissue is a group of cells having a common origin and usually performing a common function. Okay? The plant body is made up of different kinds of tissues. Let us look at it one by one now. The tissues help in body functions by allowing division of labor. For example, in leaf, various cells commonly perform the function of photosynthesis. The plant tissues can be divided into two main types. One is meristematic tissues and the second is permanent tissues. Okay? Move on to the next slide now. The growth in the plant is mainly restricted to specialized regions of active cell division called meristems. These tissue divided to form a new cells which differentiate to give rise to permanent tissues. The meristematic tissue is an undifferentiated mass of cells that is in a continuous state of division and have retained their power of divisions. Next slide. They are living and contain undifferentiated mass of rapidly dividing cells. Nucleus is large and present either in interphase or in subphases of the divisions. The shape of cell is spherical, polygonal or rectangular. Cell wall is thin with only a primary wall made up of cellulose. Secondary wall is absent. Next slide. Meristem can be classified based on their position in the plant body, function and origin. Classification based on position, meristems divided into three types. Number 1, apical meristems. Number 2, intercalary meristems. Number 3, apical meristem. The meristem which occur at tip of the root and shoots produce primary tissues are called apical meristem. The root apical meristem occupies the tip of a root while shoot apical meristem during the formation of the leaf and elongation of the stem. Some cells left behind from shoot apical meristem constitutes the auxiliary bud. Specific region of apical meristem produce dermal tissues, ground tissues and vascular tissues. Intercalary meristem. The meristem which occurs between mature tissue is known as intercalary meristem. They occur in grasses and regenerates parts removed by grazing herbivores. Both apical and intercalary meristem are primary meristem because they appear early in the life of a plant and contribute to the formation of primary plant body. Lateral meristem. The meristem that occurs in the mature regions of roots and shoots of many plants. These meristems produce woody axis. These appear later than primary meristem, so are called the secondary meristem. Lateral meristem. The meristems that occur in the mature region of root and shoots of many plants later than primary meristem, also called the secondary meristem. They are cylindrical meristems. Some example of lateral meristem are fascicular vascular cambium. I repeat, fascicular vascular cambium, interfascicular cambium, and Carb Cambium. Classification based on the functions. The meristems are also classified on the basis of their functions as protoderm. It is the outermost portion of the primary meristem, found at the apex of the stem and roots. It develops into epidermis. Procambium. It develops into primary vascular tissues. It forms the isolated strands of elongated cells very near to the central region. Ground meristem. It develops into the ground tissue. The cells are thin-walled, living and isodiametric. Classification based on origin. Primary meristems, secondary meristems. Primary meristems. These meristems are derived during the early stages. They divide rapidly and differentiate into primary permanent tissue, which make the fundamental structure of plant body. Okay, let's look at the secondary meristem. These meristem appear in later stage of development in the plant body. They lie lateral in position in both the stem and root. Some examples of secondary meristem are Carb Cambium and interfascicular cambiums. Next slide. The meristematic cells gradually differentiate and lose the ability to divide. Such cells are termed as mature or permanent cells. Simple permanent tissue. The permanent tissue having all cells similar in structure and function are called simple tissue. Parenchyma. It is a living simple permanent tissue composed of thin-walled cells. Parenchyma is also called primary tissue or ground tissue. It is mostly produced by the ground tissue. The parenchyma forms the major part of the body, major part of the plant organ. The characteristic features of the parenchyma are as follows. Number 1. Cell wall are thin-walled, less spherical and polyhedral in shape. Generally isodiametric. Cells have large vacuoles, peripheral cytoplasm with conspicuous big nucleus. The cell wall composed of cellulose, hemicellulose and pectin. The cells may be either closely packed or have small intercellular spaces. The parenchyma can be further classified as Sclerenchyma, specialized for photosynthesis. Parenchyma form a connected air system throughout the entire plant. Storage parenchyma stores sugar, protein granules, oil drops, etc. Xylem parenchyma help in conduction of water. Phloem parenchyma helps in translocation of the food. Stellate parenchyma star-shaped. Parenchymatous tissue with large air spaces. Parenchyma different functions. It helps in wound healing, grafting, and also provide buoyancy in the aquatic plants. These help in storage of the food, water and air. Parenchyma cells associated with xylem and phloem help in conduction of water and food material. The vital activities like photosynthesis, respiration and conduction are carried out inside the parenchymal tissue. Next is the collenchyma. Collenchyma is simple living mechanical tissue intercellular spaces are found to be absent. The characteristic feature of colonchimatous tissues are: number 1, it is present in layer below epidermis in dicotyledonous plants. The cells may be oval, spherical or polygonal and often contain chloroplast. It is found either as a homogeneous layer or in patches. These cells assimilate food when they contain chloroplast. On pectinization of the cell wall, there are 3 types of coloncyma: angular coloncyma, lamellar coloncyma, lacunar coloncyma. Different functions performed by coloncyma are: it provides mechanical support to the growing parts of the plant. Coloncyma cells are capable of photosynthesis as they contain chloroplast. Sclerenchyma. The sclerenchyma consists of long narrow cells with thick or lignified cell wall having a few or numerous pits. The feature of sclerenchymatous cells are: they are dead cells and do not perform any metabolic functions. They show different types of lignin deposition and also have pits. Cells are long and short, narrow, thick walled and are lignified. They possess hard and extremely thick secondary walls due to uniform deposition of lignin. The sclerenchymatous cells are 2 types: fibers, scleroid. These are thick walled, elongated and pointed cells generally occurring in groups in various parts of the plant. Scleroids. These are short or irregular, spherical, oval or cylindrical sclerenchymatous cells. The walls are very thick, irregular and luminous, very narrow. Different function of sclerenchymatous are: it provides mechanical strength and support. Surface fibers help in dispersal of seeds. Next slide. Complex permanent tissues are a group of more than one type of cells having common origin and working together as a unit. Xylem. Xylem is a complex permanent tissue which conducts water and mineral nutrients upward from the root to all aerial parts of the plant. Tracheids. These are elongated, tubular and primitive cell with tapering end walls. They are dead cells and do not contain protoplast. The inner layer of the cell wall have thickenings which vary in forms. Next slide. The tracheids conduct water and dissolved mineral elements from root to the leaves. Vessels. They are long, cylindrical tubes-like structure made up of many cells called vessel members, each with lignified wall and a large central cavity. The vessel cell are also devoid of protoplasm. Gymnosperms lack vessels in their xylem. Xylem fibers. Sclerenchymatous fibers associated with the xylem are called as Xylem fibers. The xylem fibers provide mechanical strength. Xylem Parenchyma. These cells form the only living components of the xylem. Xylem Parenchyma cells are thin walled and their cell wall are made up of cellulose. Xylem Parenchyma stores food in the form of starch or fat and other substances like tannin. The Xylem Parenchyma can be subdivided into 2 types: primary xylem. The xylem which appears first in the plant body is the primary xylem. The primary source of this xylem is the procambium. The first formed primary xylem elements are called protoxylem. The later formed primary xylem is called metaxylem. The metaxylem lies towards the periphery of the organ. This type of primary xylem is called endarch. Metaxylem lies towards the center. Such arrangements of primary xylem is called exarch. Secondary xylem. It is composed of tertiary elements, rays, fibers and interspersed axial parenchymal cells. The cell formed towards inside of the cambium tissue are called secondary xylem or wood. Phloem. Phloem is food conducting complex permanent tissue. The term phloem was coined by Nageli. In angiosperms, it is also called as BAS. Sieve elements. The sieve tube elements are long tube-like structure arranged longitudinally and are associated with the companion cell. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lack a nucleus. Golgi body and most cytosol. Sieve elements are of following two types. Sieve cell, it is a special kind of cell which possesses sieve areas in its lateral wall. Sieve tube members. In this type, the sieve areas are localized on its end walls. Sieve tube members are placed one above the other, forming a tube called sieve tubes. Next slide. The main function of sieve elements is translocation of organic solutes. Callus reappears and sometimes close the pores when solutes is less dilute, thus stopping the movement. Companion cells. These are specialized parenchymatous cells which are closely associated with the sieve tube elements. Usually a single companion cell is found associated with the sieve tubule member. The cytoplasm of sieve tube elements and companion cells are connected by thin cytoplasmic strands called plasmodesmata. Companion cells are absent in the phloem of pteridophytes and gymnosperms. Function of companion cells. The companion cells is association with phloem, parenchyma. Parenchyma plays an important role in the maintenance of pressure gradients in sieve tubules during food transport. Phloem parenchyma. The phloem parenchyma is made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through the plasmodesmatal connections which exist between the cells. The phloem parenchyma is absent in most of the monocotyledons. Functions of phloem parenchyma. They store food materials and other substances like resins, latex, mucilage. Phloem fibers. The phloem fibers or the bast fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in secondary phloem. The cell walls of phloem fibers is very thick These are much elongated, unbranched, have pointed apices. The phloem fibers of jute, flax and hemp have important economical use. The first formed primary phloem consists of narrow sieve tubules and is referred to as protophloem. Next is the tissue system. The epidermal tissue system forms the outermost covering of whole plant body. Epidermis. The epidermis is the outermost layer of the main plant body. The epidermal cells are elongated and compactly arranged to form a continuous layer. This layer is interrupted by stomata. Sometimes they are separated by intercellular spaces. It is usually single layer but is also multi-layered in the aerial parts of orchids and the leaves of Nerium and Ficus elastica. In roots, epidermis has tubular unicellular projections called root hairs. Stomata. The stomatas are opening in the epidermis of most of the aerial parts of the plants, especially the leaves. Each stomata is composed of two bean-shaped cells called gourd cells, G-U-A-R-D, gourd cells, which enclose stomatal pores. But it measures about 20 micrometers wide when fully open. In some species, the gourd cells are surrounded by subsidiary cells or axillary cells which differ morphologically from the other epidermal cells. The stomatal aperture, gourd cells and surrounding subsidiary cells are together called stomatal apparatus. The stomata are mostly found on the upper epidermis of the leaves. Stomata are following type. A. Apple type, present on the upper side of the leaf. Apple mulberry. Next is the oat type. Stomatas are present on the almost equal on two surfaces, maize and oats. Next is the potato type. They are more on the under surface. Example, cabbage, potato, beans. Water lily type. These tomatoes are more on the upper surface. Many aquatic plants. Potamogeton type. Stomato vestigials are absent. Example, Potagemitons. Epidermal appendages. The epidermis of most plants often bear outgrowth known as epidermal appendages or epidermal outgrowths. They are of the two types. Trichomes. The epidermal hair present on the stem are called as trichomes. These are epidermal outgrowth present temporarily or permanently on almost all plant parts. Emergence, prickles. They are multicellular, stiff and sharp epidermal outgrowth containing some inner tissue. Next slide. They protect the plant against excessive loss of water and grazing. They also help in climbing in some plants. Example, rose. Next slide. They serve important functions. It provides a protective covering all over the plant parts. The presence of cuticle helps in the reduction of evaporation of water. Example, epidermis. It helps in gas exchange through stomata and lenticles present on the surface. The glandular trichomes excrete various useful plant products for the plant functions. In some monocot leaves, the bulliform cells help in rolling and unrolling the leaves. Next slide. All tissues except epidermis and vascular bundles constitute the ground tissue system. In leaves, the ground tissue consists of thin-walled chloroplast containing cells called as mesophils. Like chlorophyll, it is called mesophils. Hypodermis, this is the region situated just below the epidermis and as outer region of cortex. The cortex lies between epidermis and endodermis. It consists of parenchyma, collenchyma and sclerenchyma. Cortical cells may develop chloroplast, become photosynthetic and are called as sclerenchyma. The cortex help in performing vital functions such as storage. Endodermis is the innermost layer of the cortex. It is single-layered, barrel-shaped and arranged without intercellular spaces. The cells are parenchymatous. The presence of bands of subarachnoid on the radial and transverse wall is the characteristic features. These bands are called Casparian stripes. The endodermal cells of roots usually have thick radial and inner tangential wall. These thick wall cells form a continuous rising which is interpreted as certain places by passage cells. A well-developed endodermis is present in all types of roots. Aerial stems of woody dicotyledons and gymnosperms with characteristic Casparian thickening. Next slide. The endodermis helps to control the outward movement of water and air between cones and xylem. Pericycle, it is made up of a single layer of many layers of cells present between endoderms and vascular stems. In root, pericycle comprises cells of parenchymas. Initiation of lateral roots and vascular cambium during the secondary growth take place in these cells. The pericycle is absent in roots and stems of some aquatic plants. Next is medullary rays. The medullary rays are non-vascular areas which occur between vascular bundles in dicot stems for lateral conductions. They are made up of parenchyma cells. These originates from the apical meristem. They serve the function of lateral transport. Pith. The central portion of root and stem is occupied by pith. It contains parenchymatous cells and also sclerenchymatous cells, latissiferous medullary vascular bundles. In some cases, in leaves, the ground tissue is parenchymatous and possesses chloroplast. Next slide. The main function of pith is storage of water and food material. Ground tissue of leaves. The ground tissue of pericycle is made up of parenchymatous cells with distinct intercellular space. In the lamina, the bulk of ground tissue is called mesophils, which is usually differentiated into palisade and spongy parenchyma. These cells are thin-walled and possesses chloroplast. The main function of mesophyll is in photosynthesis. Next slide. A vascular bundle is a strand of conducting tissue which is generally composed of xylem and phloem in monocots and xylem, phloem and cambium in dicots. There are three types of xylem and phloem vascular bundles: radial, conjoint and concentric. Radial, conjoint and concentric. Radial. The xylem and phloem alternate with each other by parenchymatous cells. This type of vascular bundle called radial and is found mainly in the roots. Next is conjoint. The xylem and phloem are present together in bundles or the same radius. Conjoint bundles are of two types: collateral and bicollateral. Collateral. The xylem and phloem lie together on the same radius. The xylem lies inward and the phloem outward. There are two types. In a dicot stem, the cambium is found to be present in between the xylem and phloem. Such bundles are called open. Example, sunflower, helianthus. In monocot, the cambium is absent. The vascular bundle is called as closed bundles. Example, maize. Bicollateral. This is the conjoint vascular bundle with two groups of patches of phloem, one on each side of centrally located xylem. Concentric. A vascular bundle in which one tissue is completely surrounded by the other is called concentric. The concentric bundles are of two types: amphibasal, where the phloem lies in the center and the remains completely surrounded by xylem, and the amphibrical, where xylem lies in the center and the remains completely surrounded by phloem. Next slide. The tissue organization of roots, stems and leaves can be studied better and conveniently by transverse section of mature zone of mentioned plant parts. Dichotomous root. The primary internal structure of dicot root can be studied from the transverse section of a young root of sunflower, pea or gram. The primary root is the one which has only primary permanent tissues that are formed from vegetative shoot apex. Epidermis. It forms the outermost layer in young root. It is equivalent to epidermis of stem. The stomata and the cuticle are not present in it. The cells are thin-walled and tubular. Some of the epidermis cells are prolonged to form thin-walled tubular structure called root hairs. The cells which produce root hair are called as root hair cells or trichoblast. The presence of root hair epiblemma is also called pyliferous layer or rhizodermis. Root hair have pectose layer on the outer side. This is to help them to pass into the soil space for absorption of water and mineral salts. The cells of outer epidermis shrills afterwards and become cutinized and subrised. It lies beneath the epiblemma. It consists of several layers of thin-walled parenchymatous cells with conspicuous intercellular spaces. The cells of cortex store food. It also conducts water from the epiblemma to the inner tissues. Next slide. The innermost layer of cortex is endodermis. It comprises of single layer of barrel-shaped cells without any intercellular spaces. They have characteristic band of thickening along their radical and tangential walls. These are called Casparian bands or Casparian strips. The Casparian strips are made up of suberin and lignin. These strips prevent plasmolysis of endodermal cells and do not allow wall-to-wall movement of substances between cortex or pericycle. The cells of endodermis lying opposite to the protoxylem are thin-walled to permit free passage of water and minerals from cortex into the xylem. These are called passage cells. All tissues on the inner side of the endodermis, such as pericycle, vascular strand and pith, constitute the stele pericycle. The next to endodermis lies a layer of thick-walled parenchymatous cells referred to as pericycle. The initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. Vascular strands consist of separate bundles of xylem and phloem arranged alternatively inner to the pericycle. Hence, the xylem and phloem bundles are equal in number and lie on different radii. Such vascular bundles are called radial bundles. On the basis of number of xylem bundles, the root may be diarch with 2 xylem bundles, triarch, tetrarch or pentarch, and rarely polyarchy with more than 5 xylem bundles. When the protoxylem is towards the periphery and the later formed xylem is towards the entry of the root, this kind of xylem is called exarch and is characteristics of the root. The phloem and xylem bundles are separated from each other by one or more layers of small thin-walled cells called conjunctive tissue. Later, it becomes meristematic and forms vascular cambium. Secondary growth occurs in dicot root. Pith, it is generally absent in dicot root. If present, it is small and consists of parenchymal cells. These functions to store food and waste products. Next slide. Presence of the root hair, endodermis with Casparian strips, absence of pith, radial bundle less than 8, presence of exarch xylem. Next slide. Epidermis It is the outermost thin-walled, compactly arranged layer of cells. Some of the cells give rise to root hair. The root hair are unicellular and lie in contact with soil water. In older parts, the epidermis is shed or impervious cortex. It is a broad zone of parenchyma cells. The cells are thin-walled and enclose intercellular spaces. The cortex provides for radial movement of water and minerals from epiglemma to the root interior. Endodermis, it is a single layered and made up of barrel-shaped cells which do not enclose intercellular spaces. The young endodermal cells possess an internal strip of suberin and lignin which is known as Casparian strip. Endodermal cells lying opposite the protoxylem group, however, remains in the primary stage with usual Casparian strip. These unthickened cells are called passage or transfusion cells. Steel. All tissues inside the endodermis, pericycle, vascular bundles and pith form the steel. Pericycle, it forms the outer boundary of steel. Pericycle may be uniseriate or multiseriate. The pericycle is composed of thin-walled parenchymatous cell in the young root. Later, it becomes thick-walled in many monocot roots. Vascular strand is the form of several alternate and radial xylem and phloem bundles. The xylem bundles are exarch. That is, protoxylem lies towards the outside while the metaxylem faces inwards. Due to the pressure of numerous xylem bundles and exarch conditions, the xylem of monocot is polyarchal. Protoxylem vessels are narrow while the metaxylem vessels are broad. Phloem and xylem are separate from each other by means of a narrow strip of conjunctive tissue. They provide mechanical strength on becoming sclerified. They are involved in the formation of cambium. Pith. It is large and well developed. It is large and made up of parenchymatous cells with intercellular spaces. These cells contain starch. Next slide. Presence of root hair. Endodermis with passage of cells, presence of pith, radial bundles are more than 8, xylem exarch, presence of an exodermis. Next slide. Epidermis. The outermost protective layer of stem is called epidermis. A thin layer of cuticle and may bear trichomes and a few stomata. It also helps to prevent loss of water. Cortex. This layer lies just below the epidermis and extends till endodermis. It varies. Parts are hypodermis, general cortex and endodermis. Hypodermis. It is just below the epidermis, consisting of chylomchimatous cells. The cells contain chloroplast. It provides mechanical strength to the stem. The cortex. It is located just below hypodermis and consists of few layers of parenchymatous cells. These cells have a big intercellular spaces and may contain chloroplast. Endodermis. It lies just beneath the general cortex in the form of single layer of barrel-shaped cells surrounding the stele. In sunflower, it contains starch, hence is called starch sheath. Pericycle. It exists between the endodermis and the vascular bundles. The cells are sclerenchymatous with lignified cell wall and a few parenchymatous cells dispersed in between. Each patch is associated with phloem of vascular bundles and it's called hard bast. Vascular strand. The vascular strand consists of many vascular bundles arranged in the form of a ring around a central pith and inner to pericycle. Each vascular bundle consists of phloem, xylem with a strip of cambium in between the two. The vascular bundles are thus conjoined, collateral, open with endowed protoxylem. Medullary or pith rays. These are non-vascular areas present in between the vascular bundle. The medullary rays take part in radial conduction of materials. Example, food and water. Pith or medulla, it consists of the central part of the stem. It consists of rounded, oval or polygonal parenchymatous cells. The cells store food materials and waste products also. Next slide. Occurrence of multicellular hair over epidermis. Collonchymatous hypodermis. Presence of bundle caps or sclerenchymatous pericycle over vascular bundle. Endowed xylem. Next slide. The monocot stem possesses only primary structure. The different monocot stems from outside towards inside are consist of epidermis, hypodermis, ground tissue and vascular system. Epidermis, it is single layered having stomata in it. The cells have a thick cuticle layer on the outside. Hypodermis, it is 2 to 3 layered having lignified sclerenchymatous cells present just below the epidermis. Ground tissue, it fills the whole interior of the stem containing parenchymatous cells. A number of vascular bundles are scattered in it. Vascular system. Monocot stems has scattered vascular bundles. Each vascular bundle in vascular strand is surrounded by a sheath of Sclerenchyma known as bundle sheath cells. The bundles are endowed with the protoxylem and metaxylem are arranged in the form of Y. The divergent end are occupied by toe-pitted vessel and convergent end by two-pitted vessels. On convergent end by two smaller spiral vessels lying radial in the center. A water containing cavity called as lysogynous cavity is present in association with the protoxylem. We'll move on to the next slide. Features for identification of the monocotyledon stems. Sclerenchymatous hypodermis is present. undifferentiated ground tissue. Vascular bundles scattered throughout ground tissue. Vascular bundles are conjoined, collaterals and closed. Protoxylem cavity is present. The dorsiventral leaves are generally horizontal and sunlight falls on their upper surface. Epidermis. The epidermis covers both the upper and lower surface of the leaf. Upper epidermis. It is the uppermost single layer made up of parenchymatous cell but sometimes multi-layered. Example, ficus, pipus. The outgrowth called papillae are sometimes present in epidermal cells. The stomata are usually less present in the upper surface. The stomata and chloroplast are more in numbers in lower epidermis. There is substomatal cavities present below the stomata for the gaseous exchange. Mesophyll. It is differentiated into two parts in dorsiventral leaves, that is upper palisade and lower spongy parenchyma. The spongy parenchyma lies below the palisade parenchyma and above the lower epidermis. This spongy parenchyma cells contain several chloroplasts but less than the number present in palisade cells. Vascular stems. The vascular bundles are conjoined, collateral, endarc and closed. Each bundle is surrounded by a bundle sheath of parenchymatous cells. The xylem consists of vessels or tracheae, tracheids, Xylem Parenchyma and Xylem Fibers. It is meant for the conduction of water and minerals. The phloem is made up of sieve tubules, companion cells, Phloem Parenchyma and Phloem Fibers. They surrounded by a layer of thick-walled cells arranged compactly and known as bundle sheath cell. The vascular bundles can be seen in the vein and the midrib. The size of vascular bundle varies according to the size of the veins. The veins vary in thickness in the reticulate venations. Midrib, mesophyll is absent in the region of midribs and other larger veins. Collenchyma or Sclerenchyma occur towards the two epidermal layers for providing mechanical strength. A number of vascular bundles which are embedded in a parenchymatous ground tissue. Sclerenchymatous hypodermis, monocot stems. Sclerenchymatous hypodermis is present. Undifferentiated ground tissue. Vascular bundles scattered throughout ground tissue. Vascular bundles are conjoined, collaterals and closed. Protoxylem cavity is present. The dorsiventral leaves are generally horizontal and sunlight falls on their upper surface. Epidermis. The epidermis covers both the upper and lower surface of the leaf. Upper epidermis. It is the uppermost single layer made up of parenchymatous cell but sometimes multi-layered. Example, ficus, Fibers, the outgrowth called papillae are sometimes present in epidermal cells. The stomata are usually less present in the upper surface. The stomata and chloroplast are more in numbers in lower epidermis. There is substomatal cavities present below the stomata for the gaseous exchange. Mesophyll, it is differentiated into two parts in dorsiventral leaves, that is upper palisade and lower spongy parenchyma. The spongy parenchyma lies below the palisade parenchyma and above the lower epidermis. This spongy parenchyma cells contain several chloroplasts but less than the number present in palisade cells. Vascular stems. The vascular bundles are conjoined, collateral, endarc and closed. Each bundle is surrounded by a bundle sheath of parenchymatous cells. The xylem consists of vessels or tracheae, tracheids, Xylem Parenchyma and Xylem Fibers. It is meant for the conduction of water and minerals. The phloem is made up of sieve tubules, companion cells, Phloem Parenchyma and Phloem Fibers. They surrounded by a layer of thick-walled cells arranged compactly and known as bundle sheath cell. The vascular bundles can be seen in the vein and the midrib. The size of vascular bundle varies according to the size of the veins. The veins vary in thickness in the reticulate venation. Midrib mesophile is absent in the region of midribs and other larger veins. Collenchyma or Sclerenchyma occur towards the two epidermal layers for providing mechanical strength. Number of vascular bundles which are embedded in a parenchymatous ground tissue. Next slide. Dorsiventrally flattened with stomata mostly on upper surface. Mesophyll differentiated into palisade and spongy parenchyma. Vascular bundles with colorless bundle sheath. Vascular bundles with xylem towards upper side and phloem towards lower side. Monocot leaves are generally isobilateral, but in some plants these are dorsiventral. The leaves usually show parallel venation. The various components of monocot leaves are epidermis. The leaf composes of upper and lower epidermis made up of single layered, completely arranged cells. The upper layer is covered with cuticle. In some grasses, the epidermal cells are impregnated with silica. At some places, the epidermis possesses large-sized, thin-walled, colorless, protruding cells called bulliform or motor cells. The bulliform cell may be distributed irregularly or arranged in regular patterns. Mesophylls. It is made up of chlorenchymatous, a ground tissue of leaves. It is undifferentiated. All the cells are similar. The intercellular spaces are connected, the two with substomatal cavity. Mesophyll cells contain abundant chloroplast. Hence, they are the seat of photosynthesis. Vascular bundle. A large number of vascular bundle is present. Some of them are small and some are big. Each vascular bundle is surrounded by bundle sheath of parenchymatous cells. The vascular bundles are conjoined, collateral, endarc, and closed. The xylem is present towards the upper epidermis and phloem towards the lower epidermis. Xylem and phloem elements of monocot leaves are similar to those of dicot leaves. Midrib. It is the widest part of monocot leaves. A shallow groove is present in the upper or adaxial surface, while a broad ridge is present on the abaxial surface. Features for identification of monocot leaves: undifferentiated mesophyll, presence of bundle sheath with chloroplast, vascular bundle with xylem towards upper side and phloem towards lower side. Xylem vessels are rounded. Presence of few large sized bulliform cells on the upper surface. Next slide. The growth of the root and stem in length with the help of apical meristem is called as primary growth. Most dicot plants exhibit the increase in girth. This increase is called as secondary growth. Secondary growth in dicot stem. In dicot plant, secondary growth in stem occurs both in stele and in the cortex. The tissues involved in the secondary growth are the two lateral meristem, that is Vascular Cambium and Carb Cambium. Vascular Cambium, the meristematic layer that is responsible for cutting off vascular tissues such as xylem and phloem, is called Vascular Cambium. Formation of cambium rings. The parenchyma cells of the primary medullary rays adjacent to the intravascular cambium undergo dedifferentiation and give rise to intervascular cambium. This joins the intravascular cambium of either side to form a complete ring of meristem called cambium ring. Activity of cambium ring. The cambium ring becomes active and begins to form new cell both towards and inner and the outer side. The cambium ring is made up of two types of cells: ray initials and fusiform initials. While the cells added to the outer side of cambium become elements of the secondary phloem, while the cells added by the division of ray initials to the inside as well as outside become elements of secondary medullary rays. The cambium is generally more active on the inner side than the outer. The primary and secondary phloem get gradually crushed due to the continued formation and accumulation of secondary xylem. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction. The activity of the cambium is under the control of many physiological and environmental factors. In temperate region, the climatic conditions are variable through the year. The wood formed in this season is called spring wood or early wood. In autumn, the cambium is less active and forms few xylem elements that have narrow vessels. Thus, the wood formed is called autumn wood or late wood, heartwood and sapwood. In old trees, the considerable region of secondary xylem is dark brown due to the accumulation of organic materials like tannins, resins, oils, gums, aromatic substances and essential oil in the center or innermost layers of the stem. This region comprises dead elements with highly lignified walls and is called as heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. Next slide. Carb Cambium. The stem continues to increase in girth due to the activity of Vascular Cambium. Epidermis layers are broken down and needs to be replaced to provide a new protective cell layers. Meristematic tissue, Carb Cambium or phylogens develop usually in the cortex region. Phylogens cuts off cells on the both sides. The outer cell differentiates into carb or phylogen while the inner cell differentiates into secondary cortex or phloem. Bark. Bark is a non-technical term used to describe all tissues exterior to the vascular cambium, therefore including secondary phloem, that is periderm and secondary phloem. Bark that is formed early in the season is called early soft bark, early or soft bark. Lenticles, a certain region of stem, the phylogen cuts off closely arranged parenchymatous cells on the outer side instead of carb cells. Function. The lenticles permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. Secondary growth in roots. The secondary growth in the root is the thickness due to the formation of secondary tissues by lateral meristem. It occurs by the reappearance of two types of secondary tissue, that is the secondary vascular tissues called cambium and periderm. These tissues are formed by meristem or vascular cambium and carb cambium respectively. Formation of the vascular cambium, the conjunctive parenchymal cells on the lateral sides of the phloem bundles as well as pericycle cells lying outside the protoxylem and belongs brick-shaped and meristematic. These develop into a wavy band of vascular cambium. Secondary phloem consists of sieve tubules, companion cells, phloem, parenchyma and phloem fibers. Activity of the vascular cambium. The vascular cambium derived from the pericycle gives rise to only ray initial cells. The formation of these ray cells is slower than the formation of the secondary vascular tissues. Ultimately, the cambium becomes circular. Formation of Carb Cambium. The pericycle layer after a few divisions become converted into a secondary meristem called Carb Cambium or phylogens. The cells of the phylogens divide both towards the outside as well as inside. That finishes the anatomy of plants. Bit boring. I think we just have to listen to this couple of times to know what it is. Not difficult, but it is bit boring. Anatomy of human being is excellent. You'll enjoy it. And that you'll do it when you joined MBBS. First year you'll read anatomy. You'll cut all these palms, know what are the arteries, know what are the nerves, bones, ligaments, and all those kind of things. And here and all you'll be doing all the anatomical dissections. So you are going to become a doctor, a good doctor, a best doctor.